All right, I'll let Rebecca fix that. So I am very excited to introduce Dr. Cortney Gensemer this morning and this afternoon. And I'm also very excited to welcome members of the local EDS community, both here and online. I know we have some folks joining us on Zoom. So Dr. Gensemer did her PhD in the Norris Lab at the Medical University of South Carolina. And she is currently still there as a postdoc. She led the lab in taking on a new area of research focused on Ehlers-Danlos syndromes, which she will tell you a lot more about. But for the folks in the room who are not members of the EDS community, this is a group of connective tissue disorders that are very poorly understood. And really, for those of us who are in the EDS community, if you know anything about EDS research that is going on anywhere, you have heard of Dr. Gensemer. And I essentially broke the local EDS community's Facebook group yesterday in coordinating this visit because there was that much excitement. So I'm going to go ahead and turn it over to Dr. Gensemer so she can share with you what she has been working on.
Thank you. Thank you so much for the very kind introduction. So I know the audience is like a little mix of people who are going to know a lot about EDS, but maybe find some of the science to be a little bit too much science, and people that are in the science space that will hopefully learn a little bit more outside of just the research that we're doing and some of the other things that we have going on, specifically with having patients in the lab. So as an overview, I just want to tell you a little bit about the Ehlers-Danlos syndromes and the different subtypes, and then about my journey studying HDS and bringing that into the Norris Lab, a little bit about our genetic discovery, and then how we're trying to grow EDS research beyond just our work in one single lab, some of our patient scientist initiatives, and then kind of going beyond just what we're doing in the lab to transform clinical care for this disease, which is desperately needed. So the Ehlers-Danlos syndromes are a group of 14 different connective tissue disorders. They have variable presentation, different genetic causes, but as a general rule, they share the features of joint hypermobility, so joints that go beyond the normal range of motion, often skin hyperextensibility, and tissue fragility. You'll see a lot of abnormal scarring, wound healing changes. You'll also see types of spinal instability, a lot of things that can often be brushed off as sort of individual problems until you start looking at the bigger clinical picture and connecting the dots. This is really viewed as a connective tissue disorder or an extracellular matrix collagen disorder. And we're really going to focus on the hypermobile subtype of EDS called HEDS. On the left here, you see the general sort of signs and symptoms of HEDS. So a lot of the things I mentioned, hypermobility, that can lead to joint subluxations and dislocations, things like hernias and pelvic prolapse, which are kind of these more systemic connective tissue problems, issues with the skin, as well as the cardiovascular system. But we also see a lot of comorbidities in these patients that aren't always considered part of the disease. So things like dysautonomia and POTS, which has gotten a lot of attention since COVID. We see a lot of things like mast cell activation, overly active mast cells and immune responses, neurological issues like spinal instability, migraines, tethered cord syndrome. And it's actually the most common type of EDS. Previous estimates used to say it was around 1 in 5,000 people. We now know it's much more common than that. It's estimated to affect 1 in 500. So although it's considered a rare disease, that actually makes it really not a rare disease. It presents in this spectrum of severity. So not every person is going to have every single one of these things pictured here. Everyone can be very different in terms of how severe it is and what specific manifestations they have. There's no specific treatment options except really managing symptoms. So that can be physical therapy, pain management, sometimes surgical intervention to correct issues. It's often misdiagnosed and underdiagnosed. It goes undiagnosed for a lot of patients for a really long time. There's a lack of clinical awareness and education around the disease. There's no clear diagnostic test or genetic marker. There's just a set of clinical criteria that you sort of have to hope you walk into a doctor's office who knows about it and decides to pull it out and go through it with you. What's interesting is a majority of those affected are women, or women just tend to be more severely affected than men. So as I mentioned, this is kind of like the spectrum of the phenotype. What I do want to say is that a lot of times we see these worsening of symptoms around the time of puberty, which makes diagnosis even harder. You have a lot of young women who are going to the doctor with a lot of complaints that often aren't taken seriously. And then a lot of patients also report sort of triggers in which their symptoms became more obvious or more severe. So this is just an example of 3 patients and how they might present differently. But despite this, it is an inherited condition. We see it running in families. However, we often see different levels of severity throughout a family. Before I talk a little bit about our research, I do want to share my journey in science, and this is a really crucial part of the story and just sort of my journey in general. So I grew up in a super athletic family, playing sports my entire life. It was super normal to think that I was sore all the time or experience a lot of pain. I got excited about research in undergrad. I started doing research, learning about PhD programs. And then I finished my degree in 2018. I went to MUSC for graduate school, finished grad school, and I stayed on as a postdoc. But what you don't see in all of these pictures is all of the things that happened in between and all of the health issues that I dealt with because of hypermobile EDS. And I was diagnosed while I was in college. I've had more than 10 major surgeries. I've been hospitalized many times. I am pretty much in a state of chronic pain all the time, even though it might not be obvious to other people. But I didn't know my whole life. And because I didn't know, I made a lot of lifestyle choices like athletics and other things that definitely damaged my joints more than if I had been diagnosed at a younger age. That being said, I wouldn't change any of it because this experience has shaped my research. And I think it's also really changed my perspective and a lot of other people's on what it means to be a scientist and a scientist with a disability. So I'm going to share this video with you that MUSC actually made to highlight what we're doing. And this kind of gives a little bit more context to the story.
From a young age, I've always loved science. I was interested in pursuing a career in research. When I got into MUSC for graduate school, I started my PhD and I didn't really know what I wanted to study. I met Dr. Norris.
So she asked me what I did, and I said I work on heart valve disease. And she revealed that she had Ehlers-Danlos syndrome.
I was diagnosed with HEDS when I was around 19 years old, but there's no direct treatment and really not a lot of research going on into why this is happening or what we can do about it. Most days I have been able to manage it, but I'd be lying if I didn't say I'm in pain basically every second of every day.
I asked her what the gene was, and she said nobody knows.
He was like, well, do you want to try and find the gene? Find the gene, we can do that. We started with a large family that had HEDS to kind of narrow down and find a genetic cause. From that, we were able to have a small list of genes that we then narrowed down to one causative gene for HEDS. With the genetic registry that we developed, we found this mutation in other patients and other families as well. This was the first gene discovery for HEDS.
MUSC really enabled us to become a world-leading institution with a focus on Ehlers-Danlos syndrome.
Even as a student, I always had a seat at the table. To have leadership and administration listen to someone who was, just a PhD student is really rare and unique. It takes a long time to make a scientific discovery. This process is slow, so to have such a big discovery so early in my scientific career is kind of unheard of. It made me really excited for every next step along the way.
So when we started this, my mentor, as he mentioned, really didn't study EDS. And for me, it was something that I was really passionate about, but not something I went to grad school planning to do. I knew that there were a lot of hurdles in EDS research. When I was diagnosed, I was, of course, reading the papers, trying to understand the science, looking at who were the leaders in this space. There weren't a lot of them. There was not a lot of funding. And there was really just this total lack of baseline knowledge about what this disease really is. So for us, it was really hard to know what to do. Like, where do you start when nothing is really known? My mentor was willing to take a chance on this, and he had worked on similar studies in the past with cardiovascular diseases. So we just hoped that maybe we would get lucky in this process. So I want to show this cycle of discovery that we use to talk about work that we do in the lab, because it really is kind of our overall goal and what we hope to do. As researchers, we collaborate with clinicians at MUSC and other institutions to develop patient registries, recruit patients, collect things like tissue, DNA, serum, and plasma for our studies. And then we take these samples and we can use that to inform animal models, do genetic studies, biomarker studies, and take this information. And then ultimately the goal is to end up here, right? To come up with ways to better diagnose this condition, treat it, and have that go back to patients. Now that's not that straightforward. That process takes a really long time. But to get started, we developed a genetic registry for HEDS. So as I mentioned, this condition is diagnosed based on a set of clinical criteria. So these are the two main parts of the criteria where they look to see if you have generalized joint hypermobility based on something called the Beighton score. And then they also have a second set of criteria that really just looks at all of these other things that are more indicative of like a systemic disease. So do you have mitral valve prolapse? Do you have atrophic scarring? Do you have a history of pelvic floor prolapse? Things like that. And if you check enough things on this list of criteria, a physician will make a diagnosis of HEDS. So these patients were all clinically diagnosed. We also recruited family members that were affected and unaffected in the same families, and got saliva samples for DNA, collected basic demographics, and just some basic phenotype information, like what symptoms do you have? How does this affect you, to kind of understand the patients that we were recruiting. And we started our first genetic study with a large family that we identified with HEDS. So you can see those with a clinical diagnosis in black circles and squares, and then those that are shown in gray are what we deemed, or the clinician we were working with deemed, as probable HEDS. So these individuals, these two, right, the 3-3 and 2-3, I just realized I can actually write with this. So these two were older at the time of consent, and so with age, the joints tend to stiffen. They were kind of put in that probable group just based on a history that was super consistent with HEDS. You can see this one that has probable HEDS has tons of things listed, ankle instability, the one above it, repeated meniscus repairs on EG, multiple ankle reconstructions, severe lumbar spine surgeries, torn ligaments in the elbow, all of these things that really met an HEDS phenotype but couldn't be evaluated based on that criteria. And then the youngest individual in the pedigree was just 13 at the time, had a lot of symptoms, but the provider didn't feel fully confident in saying they definitely had HEDS. And this is fairly common for younger people. So we, in this case, categorized all of these people as being seen as affected. And we went ahead and we chose the two most distantly related individuals, so this one and this one right here, to initially do whole exome sequencing on. And we found 48,000 shared variants among those two individuals, but only 6 of them that were rare and potentially damaging once we went through the rest of the pedigree and sequenced those 6. There was only 1 variant that segregated with the individuals who were affected. And so that was in a gene called KLK15. This is not something that was previously known to be involved in connective tissue disease or Ehlers-Danlos syndrome. It was a glycine to an aspartic acid mutation. It had an uncommon frequency. I would say rare. It's debatable if that's considered a rare mutation, but it's rare enough in the general population for a disease like this that it made sense. It was predicted to be damaging by many different pathogenicity predictors. And then we also found it in a second small family. So this was just an affected parent and child, but again, the same mutation. So what are the kallikrein-related peptidases? So these are a very large cluster of trypsin-like serine proteases. These are enzymes that have a catalytic site that can cleave different substrates. They're all located right in a row in the same locus, and then biologically have very similar functions. We know that they are hormonally regulated, which was interesting to us. They are involved in connective tissue biology, of course, involved in cancer, but also involved in things like immune cell function and blood pressure regulation. So KLK15, of course, was the one with the least known about it. When we first started looking, there were a handful of papers, and no one knew much about what it did or where it was. So we asked the question of, okay, is it even expressed in relevant connective tissues? Does this make sense in the context of a connective tissue disease? Initially, we did searches in common databases that people will look at expression data in, and found that those databases, of course, didn't include a lot of connective tissues aside from maybe the skin. So we found a different database that integrates lots of different single cell and bulk seq data, has its own sort of proprietary scoring algorithm to compare expression and all these different from all these different types of data sets. And from there, we saw that KLK15 was actually very highly expressed in tissues like the tendon and the skin and lots of things that kind of made sense with this disease. But we wanted to validate this ourselves too. So these are some RT-PCR from mouse skin and Achilles tendon. We included the thymus as a positive control as it was known to be expressed there in mice. So we saw KLK15 expression in these two connective tissues, which were really the main connective tissues that we can get from a mouse model. And then in humans, we looked at this in ACL tissue as well as dermal fibroblasts. So this was really encouraging to kind of further understand, okay, it's expressed where it is, it tracks in the family, what really are the consequences of this mutation? So we did some initial experiments to sort of look at how the mutation might affect what the enzyme's doing. We didn't know what to expect, but we looked at the enzyme intracellularly and extracellularly. And what we saw is with the wild type, we have wild type KLK15 here when it was transfected inside the cell. And you can see secreted into the media down here. When we transfected the mutant, so these have two different tags. The mutant was tagged with an HA and the wild type with a his tag. When we transfected the mutant, we saw that it also got secreted, although maybe not quite as much. But we weren't as interested in that as we were in really what happens when you have them together. All of these patients are heterozygous. There's really no homozygous individuals. So when we put them together, we actually saw that the wild type got stuck inside the cell when it was transfected with the mutant enzyme. And we saw less of the wild type secreted. So really this was having, to us, what looked like a dominant negative mechanism and effect on the wild type. And this for us was kind of enough to justify going forward and looking at it in an animal model to see what was going on. The best way to really validate the genetic finding would be to see if mice with the same mutation have a connective tissue phenotype. So we used CRISPR-Cas9 to knock in this mutation into the mice. They do have 2 fewer amino acids, so it is going to be G224D in these mice. And then we said, okay, we're going to phenotype them for HEDS. But we realized pretty quickly that this criteria is not easy to apply to a mouse. Mice are actually really hypermobile, and their feet like bend all the way backwards and can touch their shins just in a normal mouse. So trying to understand how to see if these mice had hypermobility was going to be a little bit more specific than that. So we took Achilles tendons from these mice, and we worked with some bioengineers at MUSC who were able to do tensile testing of the Achilles tendons to measure the elastic properties. So these are tendons from the wild type mutant mice. There were no initial differences, same size, same length, everything like that. And using a tensile testing system, basically what happens is the Achilles tendon is put between these clamps here, and a load is used to stretch the tendon basically until it breaks. But from that, you get all of these data points that give you information on the elastic properties of the tissue. So what we saw with this is really an obvious change in this toe region specifically. And what that toe region of the stress strain curve corresponds to is actually the initial uncrimping of collagen fibrils. So you have these collagen fibrils, and before they start really getting stretched, they kind of uncrimp at first. That transition point that's being pointed to right here is when that toe region sort of stops. And so when we compared the tendons at this point, we saw that the transition displacement, so that initial stretching that the tendons could do, was increased in mice with the mutation. The amount of strain at that point was increased, and the toe modulus, so the elastic modulus at that point, so a lower modulus is more consistent with a more elastic tissue, was decreased in mutant mice as well. So we saw that, but we wanted to know what about the actual structure of collagen? How is that different in these mice? So we were able to use the transmission electron microscope at the University of South Carolina to actually look at the ultrastructure of collagen in these tendons. So in a normal tissue that's full of collagen, you generally see variability in fibril diameter. That's very normal. But the variability that we saw in these mutant mice involved a lot more of these smaller collagen fibrils. And so we quantified very, very, very many fibrils from wild type mutant mice, and you can see a decrease in just the overall collagen fibril diameter. And then when we bend these in different increments, you can just obviously see that leftward shift of the purple of these mutant mice having smaller collagen fibrils. That was great, but this is one tissue type. We know that this is a systemic condition. So we asked the question, do they have other connective tissue phenotypes? And kind of looking back at that list of criteria, we were like, what are other things that we can look at? And what are things that these families had that might be relevant in the mice? So being a lab that has historically studied the heart, that was kind of an easy target for us and something we had the tools to do. So what we saw here when we looked at the hearts of these mice, so this is a MOVATS pentachrome stain. This is the anterior leaflet of the mitral valve, posterior leaflet of the mitral valve, and then the aortic valve as well. And so typically what you want to see with valves is kind of a thinner structure that can open and close, not prolapse up into the atrium. And what we saw here with these mice is that where these arrowheads are, we saw myxomatous degeneration. So basically the tissue of the valve is getting thickened and replaced by different connective tissue, leading to typically prolapse, and also changes in the hinge of the aortic valve as well. And so to validate this further, similarly to the tendon, we saw both the change by looking at the ultrastructural, but also these functional changes when stretching the tendons. We wanted to see if functionally these mice had prolapse. So we performed echoes on mice with the mutation and controls. It's really tough to see echoes on a still image from mouse, but you can see the outline of the two leaflets of the valve. Typically what you want to see is that they seal tightly and that they're not bulging up into the atrium. And here with the mutant mice, you can see that bulging happening. We did see regurgitation in some of the mice, but it is much harder to detect in a mouse model than in people. And then we also looked at the aortic diameter. So in hypermobile EDS, you sometimes see aortic dilation. It's not as common as other connective tissue diseases like Marfan syndrome or vascular EDS. We did see a trend towards increasing aortic diameters, but it wasn't significant, and we really didn't necessarily expect it to be, but we took a look at that as well. So this was kind of our initial phenotypic validation of these mice. And to really summarize it, we found this mutation in KLK15. It tracked with the disease in families. We confirmed expression in connective tissue. We saw evidence of in vitro pathogenicity, and then we validated connective tissue defects in the mice. But that's only 2 families that we know this is not a rare disease, and we want to know what's going on in the broader HEDS population. So we got interested in the other KLKs and looking beyond just KLK15. They're all in one continuous locus. There's also this overlap of how KLKs sort of regulate each other. And so theoretically, if you were to mess up one of these somewhere, they could affect the downstream KLKs and their processes. So we expanded that spectrum that we were looking at and just decided to see, do we have mutations in other KLKs? If we do, is there maybe any evidence that something is going on here? So we started with 206 patients that had whole exome sequencing. These were all completely unrelated individuals. We excluded any family members. We excluded any other connective tissue diseases in these patients. And we went ahead and applied the same filtering that we used previously to make sure that they were rare and predicted to be damaging. And we identified 76 variants, 48 of which were unique, so some of these appeared more than once, in 65 patients with HEDS. And then we took a control cohort. This was from Nomad, and we applied the same filtering for quality and things like that. And we went ahead and did a comparison with our genetic colleagues who performed a gene-based burden test. And so really this just takes all the variants from our cohort, compares them to a large population of controls and says, is there enrichment in having these mutations in patients with HDS? And we saw that there was. The entire locus was very significant, and then also breaking it down by the gene level, we saw enrichment in actually every KLK gene except for KLK 4, 9, and 11. So that actually showed us that about 32% of HDS patients had at least one rare variant that was predicted to be damaging in a KLK gene. But that still kind of leaves us with like, how are we getting from there to a collagen problem? And that's really where the bulk of my work has been focused for the last year or so. We performed a yeast-2 hybrid screen. So this is a way of identifying protein interactions. Basically, if you have an interaction between something, you will get transcription of a reporter gene that can then be sequenced, and you can identify interacting partners. So KLK15 in this case was our bait, and we used a fibroblast library to look at connective tissue interactions as our prey. And our initial hits of interest was much longer than this list, but these were the 4 that in terms of connective tissue were really interesting to us. Fibronectin is an important scaffold for the extracellular matrix. It also helps organize and assemble collagen. And then lysyl oxidase is an enzyme involved in collagen cross-linking, so controlling the size of collagen fibrils, which made sense in what we were looking at. So we've gone ahead and tried to validate these interactions. We have validated an interaction by co-IP with KLK15 and fibronectin only extracellularly. So here when we have KLK15, and pull down for HA, we don't see an interaction in the cell lysate, but in the media, we do. And we actually saw the same thing with LOX. I don't have that data in here. But we've confirmed both of these interactions so far. We're looking into collagen 6 as well. But our future studies will really focus on how these interactions are important and how maybe the dominant negative mechanism or the specific mutation interacts or interferes with those interactions. As well as if other KLK mutations or other KLK genes have similar interactions in the extracellular matrix. So to really summarize this part of the talk, we know that there's this broad involvement of the KLK gene family or suspected broad involvement in HDS. We have studies ongoing to understand these mechanisms, but I also really want to point out that HDS is definitely complicated and polygenic. This is maybe only one small percentage of what's going on. I know the fun video MUSC made likes to make it sound like we found the gene or the solution. Certainly not that simple. There's a lot going on. Most of this data that I just talked about is out as a preprint right now if you want to check it out and kind of look at it a little bit more closely. We've had quite a fun experience trying to get this published with a couple of years at some really great journals and lots of reviews that unfortunately ended with rejections. So we're in that resubmission process right now. But I'm glad that the preprint is out and people have access to that information. Now, all of that being said, what about the other 2/3 of HDS patients? So I have spent some time working with colleagues learning about population genetics. I am not trained in genetics very much at all. I am a cell and molecular biologist. So this has been a really cool opportunity to get to work on a GWAS with people who really know what they're doing in this space. So a GWAS is a genome-wide association study, for those who don't know. Basically, it's a way to identify common variants that might be involved in a disease. So we have our large registry. We've now have over 3,000 DNA samples from patients with HDS. It's probably the largest, if not the only, big collection of HDS samples that exists. And we have a clinical questionnaire that's paired with. And so one of the really cool things that this allows us to do is actually compare specific clinical phenotypes to loci of interest from our GWAS and see if there are actually different genetic hits of interest that correlate to specific phenotypes. And maybe this complex sort of catch-all of a connective tissue disease will be broken down a bit more. So right now, we just completed our second cohort, and this data is being finalized so we can start some biological validation in the lab, which is really where my role is going to fill in a bit more in understanding this. And we're also working on some biomarker studies. So we recently got serum and plasma samples from HDS patients and controls and are working with some people in our proteomics core at MUSC to look at any potential biomarkers in this data. Our first cohort was very small, but we have identified some things that seem promising and are kind of in that validation step right now. One of the biggest things I think we can do to make an immediate impact on patients is have a diagnostic tool, shorten that timeline to diagnosis, and have an easy way to say, like, here's a blood test, you know you have it, and let's start getting you appropriate care. So through our GWAS and through the biomarker data, we're hoping to get some new insights into the clinical picture. And I do want to highlight our recent clinical paper that we published. So this was led by a PhD student in our lab, Taylor, who did a lot of awesome work on this. Basically, when we started our registry, we thought we might get a few patients or a few families. When our EDS registry first hit an EDS Facebook group, we had 1,000 patients express interest in participating overnight. So we quickly put together a clinical coordinating team. We were so grateful for everyone wanting to be involved. But looking back, I think we could have maybe expanded this, but we had an initial sort of questionnaire. So when you were consented and provided your DNA sample, there were a few questions that you answered. Do you have X, Y, Z symptoms? Check yes or check no. I mean, that was all we thought we wanted to collect. And so after we got over 2,000 patients, we thought maybe this is actually something that's useful to publish and get out there. So through this information that we got, we were able to do a cluster analysis and see if there were actually sort of subclusters of clinical manifestations in HDS. And what Taylor found from this is that there were 3 clusters that sort of fell out of this data. What was interesting is when we looked at the Beighton score and measuring for hypermobility, the clusters fully disappeared. So these clusters were truly based just on the phenotypes and comorbid conditions that patients reported. With the first cluster, we really just saw this higher prevalence of a lot of these diagnostic phenotypes. So the things that were maybe typical EDS, maybe patients even got diagnosed more easily, that kind of fit into that first cluster. Cluster 2 had a lower prevalence of most phenotypes. However, I'm hesitant to say they were less severely affected, as we really didn't ask that question. And cluster 3 really had this interesting phenotype presentation where they had the highest rates of all of the neurological comorbidities we asked about and the higher rate of mast cell activation syndrome, as well as the highest number of conditions. So we have plans in place to try and merge this. We've recently shared all of our cluster data identifiers with the collaborators that we've done our GWAS with to see if there is anything that sort of aligns with specific clusters from that data. So to summarize these ongoing studies, we now have a GWAS going on over 2,000 HDS patients. Our serum and plasma proteomics will hopefully expand and maybe identify potential biomarkers for HDS. And then we've really seen some interesting clinical insights from our survey. We have an additional survey that was created by a postdoc in our lab who was interested in trying to understand this clinical picture even more. And we had over 9,000 people fill out the survey. So she's in the process of analyzing that. It's much more in depth. It really tried to take an unbiased look at HDS without just saying, okay, we know these are the phenotypes, but instead kind of letting the patient community report to us what the disease really is because it's so poorly defined. So that will hopefully be published in the near future. And then we're also trying to really understand the involvement of the KLKs and other genes in these comorbidities, such as POTS and MCAS that happen in HDS. So one of the things that I think is really unique about our lab is my mentor has become very committed to growing EDS research, not just in what we're doing, but globally, and especially in the US. There's a huge issue with access to care. Our recent survey that I just mentioned that's not published yet, we see that it takes on average 22 years from symptom onset to EDS diagnosis. Disease management is a big problem. Patients saw on average 7 different specialists in a year. 39% have had at least one inpatient hospital stay. 30% of patients have traveled more than 300 miles for care. 76% say they have paid out of pocket for specialists not covered by their health insurance. And 45% of patients spend more than 5 hours a week coordinating their own medical care. And then, of course, there's lack of research. So there's very few researchers focused on HEDS or EDS in general. Is it because of lack of interest, lack of funding, or because nothing's known? I'm not totally sure, but we really want to change this. One of the things that we've looked at is really the funding gap. So if you look up hypermobile EDS on NIH Reporter, there's 13 projects that have ever been funded that mention HEDS. As you see, there's only, at least in the initial 5 that are listed, only one that actually mentions it by name, and that's because it's mine. So there's really a huge issue with either people aren't submitting grants on it, which could totally be true, or they're not getting funded. And there's certainly not any R1s focused on hypermobile EDS at all. So as a comparison, I wanted to look at another condition that has like somewhat of a similar prevalence and just see what's going on. So I looked at multiple sclerosis and saw that there were 23,000 projects ever funded on it, which is great. I'm not saying that shouldn't happen, that absolutely should, but the comparison is really striking. And when you look at the actual amount of money that's been spent, so nothing was ever funded that mentioned HEDS before 2007. And the total amount of money has been for $1 million versus $9 billion. So we really want to change this. One of our efforts to do this is to provide tools to researchers interested in getting started in this space. There are a lot of connective tissue biologists out there that just don't study connective tissue in the context of EDS. So we had a very generous donor who wanted to help grow EDS research, and she gave us the money to generate an animal model for every type of EDS, and these are actually available to researchers for free. So our progress with that so far is that we have almost all of them validated and ready to actually be shipped to researchers. We're really trying to promote this. It's not just for the hypermobile type, but even the rare types of EDS that also don't get any research attention. So if anyone knows anyone who might be interested, please share this information with them, especially as they get new students, maybe someone's interested in studying EDS as a project. It's a great way to get started. But at the end of the day, there still needs to be funding in place. So we've worked with leadership at MUSC and in the state of South Carolina to actually get specific wording into the Senate report for labor, health, and human services for 2025 to hopefully encourage the NIH to support HEDS related research at academic medical centers where HEDS patients are being treated. It was really, really cool to see our own wording actually make it into this book. I have no idea if it's going to make an impact, but I'm hoping that it's discussed. If NIH could make this an initiative, it would really encourage researchers to apply to the grants if there was funding set aside for this disease. But the most important thing that I really want to talk about is that none of this would have been possible without patient involvement. And it's not just me. There have been many other patients now that have worked with the lab. What we've learned from this is that patient representation in research and in medicine is honestly so crucial. I think if you don't have patients represented in what you're doing, if you're doing biomedical research, your work just really doesn't serve as much purpose, whether it's patients being a part of a grant review process for an organization or patients actually working hands-on in the lab. You want to know that the work you're doing is actually making an impact. And my perspective as a patient has really shown us the power of patient involvement in research. So we wanted to expand this, and that really led us to develop an HEDS intern program in the lab. So the goal was really to initially focus on undergrads who actually had EDS themselves, that wanted to do research in the space, maybe wanted to go on to medical school or graduate school and study this. We also wanted to build this network and community of people who understand this disease, who go on to all these different careers and hopefully collaborate and work together in the future. But a big part of it has also become disability advocacy, making the lab accessible, providing an accommodating and accessible environment for students to work in. The number of times that I've been told by an undergrad, I didn't think I could be a scientist because I use a mobility aid, or labs aren't accessible, research isn't accessible. That's not wrong. Research is historically not accessible, but we're really trying to change that culture, not just in our lab, but also the entire MUSC campus. And then also prepare them for their careers. So under other undergrad research programs, they get mentorship, they learn about our med school, our PhD programs, They shadow physicians at MUSC, things like that. So our first year really started because we had a private donor give a donation, and that was when the idea came up, and we said, okay, we'll try and take a couple interns this summer. So we had 25 applicants, we took 4 interns. We had another foundation reach out that really loved the idea. And that was for year 2. They funded us for year 2, 3, and 4. They actually very kindly insisted that when we got the donation, we wanted to name the program after them. And they insisted on actually naming it after me. So it's now the Gensemer HEDS intern program. And we have seen a growth in the number of applicants every year. This is funding TBD for year 5, but we actually just got funding for year 5, and also probably year 6. So the program will be continuing to grow. But during that time, another foundation reached out and said, we want you to make this bigger. We only have the capacity to take so many students in the lab in the summer. You only have so many mentors available to train them. So that led to the development of our Visiting Scholars program. And the idea for this is really to get young people who are interested in science excited and want to come out for a few days, see the lab, get some exposure, maybe figure out what they want to do. So we have this program ongoing year round. I think we have 20-some people coming in the next couple months over different days. And not only do they get to be a part of that, but we bring them on the same days as each other. So we have a few students, and for some of them, they've never met someone else with EDS. And so it's a really impactful and empowering experience for them. And so our initial goal was to say, okay, we'll have trained about 55 patient scientists within 6 years. We've already surpassed that goal in 5 years. So our new goal is to train over 100 patient scientists within 6 years. And I think that's definitely going to happen. Our past interns have now gone on to graduate school and medical school. From our very first cohort, we have a 2nd-year MD PhD student at MUSC who's actually planning to come join the lab for the PhD portion and continue to work on this stuff. We have graduate students at multiple different PhD programs now across the US. But the biggest thing and the biggest takeaway that they have given back to us is that their success wouldn't have been possible without the accommodations and this inclusive environment. And what we've really seen is that supporting students with disabilities has allowed them to thrive so much. We have done a lot of things in the lab that are honestly not that hard to do, that make it sort of this safe space. And we try to provide accommodations in more of a way that they don't have to ask for it. It's just, we have a handicap accessible door that you wave to open, and we lower some of our benches so that you can sit at them. And we just have that set up because putting that burden on someone with a disability does make it challenging. People are scared to ask, they're embarrassed, they don't want that attention on themselves. And so doing whatever we can ahead of time, but also if there are specific requests, of course, trying to meet those in any way that we can. We have ergonomic pipettes. We actually get these special low dermatitis potential gloves and all sorts of things that really just make it normal in the lab to be accommodating. And one of the things I had a spinal fusion at one point during graduate school, and so I was wearing a neck brace to lab. And we had one of our high school interns who was also had EDS, and she told me that she was now not scared to wear her neck brace to high school because she saw me doing it at work. And so normalizing that culture in the lab has been really impactful for people and makes them feel like they can be successful because they don't have to try to hide that part of themselves. So I'm throwing this out there. If anyone here is interested or knows anyone who might be interested, our summer intern program applications will open in November. And then I think our Visiting Scholars is constantly kind of open and a rolling process. So if you're interested in that as well, you can always apply. So the last thing I want to talk about is how we can really make an immediate impact at MUSC and something that we're doing that I never, ever would have thought would happen when I was a graduate student. And that is really putting together an EDS institute at MUSC. That includes research, education, and clinical care focused on EDS. During my, I think, 3rd or 4th year of graduate school, we started having conversations with clinicians about how they struggle with managing these patients. So a patient might come in, we have a neurosurgeon who sees a lot of EDS cases, and he fixes the neurological problem, but then they're like, well, I have this, what do I do? He has no idea where to send them. And patients start patchworking care from all over the country. So this lack of centralized care is a huge problem. And it also limits the potential collaboration of physicians and limits the ability to do exciting translational research. So we really saw patients coming to MUSC for one specialty and then leaving. We're like, we need to change this. So think of a cancer institute, right? You can come in, you can have multiple appointments scheduled with different specialists. You have a patient care navigator who helps you with everything. We want it to be like that. So we're working on developing this institute. We're working on pairing it with education, not just our EDS intern program, but healthcare providers, nursing, all of these different things, both students at the institution and also current employees. And then the overall outcome ideally would be decrease healthcare burden, improve quality of life, and also will really further research efforts. We have a lot of IRBs in place right now, but if the patients aren't coming to MUSC for their care, we can't get tissue samples and other things like that to grow the research. So right now we have, hopefully we will be able to soon say we have a medical director. We have job offer out for a medical director, and that person will come in and then build out the other clinical specialties that are needed to get this started. And then also recruit more faculty to do EDS research because it definitely needs to be more than just this one lab. But also this is needed everywhere, not just at MUSC. When you have a disease that affects 1 in 500 people, you should be seeing experts and centers all across the country. And it's starting to happen. I do know of other places that are starting to do this. And I think that it's with how much we're seeing this diagnosed, I think that we're going to need them even more. I know that it can be really challenging for families when you have a condition that affects multiple people in your household. So you're not just, looking for care for you, but you're looking for care for your kids too. And so to be able to travel together and see all the doctors you need in one place would make a huge difference for these patients. So really just to summarize the last few things, there are a lot of research gaps that we need to address. And I'm hoping that NIH support will really help to do this. I also want to emphasize that patients should be a part of the research progress. This is not just EDS, but this is so many other diseases. Patients have really cool, unique insights. They are super passionate about solving their own disease. I mean, the things that I learn from the interns every summer are incredible. And then by pairing this research with education and clinical care, I really think we can totally transform the way this is studied and treated and viewed as, or often misviewed as this rare disease. So I do want to acknowledge a bunch of really great people at MUSC and beyond. Everyone with an asterisk has been a huge part of this project that I definitely want to point out. My mentor, Dr. Norris, none of this would have happened if he didn't just say, oh, you want to find the gene? Like it was a casual conversation. That happened the first time I met him too. It was not like once I had been in the lab, it was our very first conversation. I didn't know he was completely crazy or if it was going to be like something that would change my life. It was definitely the second one. The first one, maybe a little bit too, Our intern program, all the interns, if I tried to list them all from the last 4 years, there would be very many names. But all the interns that have contributed to this, collaborators at MUSC, our transgenic and genome editing core that made all these mouse models possible, our colleagues in Paris, at Mount Sinai and Harvard as well. And then the South Carolina Clinical and Translational Research Institute was basically handed an IRB with 1,000 patients to consent overnight. So they deserve a huge shout out for handling all of that. And then of course, all of our donors who have made this possible and all of the EDS patients who have participated in this research. We obviously couldn't do it without them being involved. So we have a really awesome website that Molly in our lab has put together. So if you want to learn more about any of our projects, intern program applications, anything like that, it's all on there. And then also here's my email if anyone has any questions, wants to talk about anything. I also spend a lot of time on social media making content related to EDS. I share research and work from others in the EDS community, break down scientific topics, all those things on social media, and it's Cortney Science if you want to follow me there. So I will leave it with that and let you ask me any questions you might have.